Good afternoon, folks. Hope this finds everybody well. This is the beginning of our section of the course that will deal with the Kingdom Animalia, which is going to dominate most of our attention for the remainder of this class. For those of you who are my students, strongly recommend you go on Blackboard and get yourself a copy of my handout on the origins and general features of animals for following along with this lesson. So, Kingdom Animalia. This is the group of living things that we are most familiar with because this is the group that we are a part of, although humanity has long been in the habit of thinking of ourselves as being somehow separate from the animals. We are, by all classification schemes that you can use today, animals. It's only our real hubris, honestly, that makes us think of ourselves as something separate and different. Now, classification of animals goes back a long way. It's been done all the way back to the time of Aristotle, whenever he realized that animals represented a distinct group of life apart from other living things. Of course, Aristotle put humans outside the rest of animals, but he's often been called the first zoologist. Kingdom Animalia, as we understand it today, is an incredibly diverse group of organisms, encompassing approximately 1.3 million identified species of life, with possibly as many as 10 to 20 million living species out there. Most zoologists agree that we've probably only discovered about 10% of the total diversity of all animals that are presently living, and that may only represent 10% of the diversity of all animals that have ever lived. The actual number of animal species that has ever occupied this earth probably goes somewhere between 100 and 200 million distinct varieties. So animals represent a tremendous group of living things. They're extraordinarily diverse. They occupy basically every habitat on the planet that supports multicellular life. And they range from simple things like sponges up to extraordinary complex vertebrates like ourselves. So we've got this massive group of living things. What makes an animal an animal? This is a not easy question to answer. Biology has long labored with how to classify the animals, and this has been muddied by the existence of multicellular protists that have animal-like features, and by certain animals like sponges that seem an awful lot more like plants. So our definition of what makes an organism an animal has slowly evolved over time, and has some fuzzy areas at the edges. As you saw when we discussed plants, there is still considerable debate over whether to classify an algae as an animal, or as a plant or as a protist. Likewise, there are certain simple multicellular protists that there's debate about whether or not they could be more meaningfully classified as animals. And as we're going to get into in the near future, there's considerable debate over exactly when the Kingdom Animalia even began, when it even branched off from the other groups of multicellular life. So we need some working definitions. So how do we classify this thing we call animals today? Let's go with the basics. First off, basic nutritional mode. This is arguably the most important distinguishing feature of the Kingdom Animalia that sets it apart from the Kingdom Plantae, in that animals are heterotrophs. Heterotrophs, heterotrophs. Translate that, troph to eat or to feed, hetero, other, feeds on others. Heterotrophs are organisms that require external food. In other words, you as an animal, unlike a plant, cannot synthesize your own food molecules. You are a heterotroph. You must take in external food from your environments. That means you are either ingesting other organisms, whether those are plants, fungi, or other animals, or you are in some way sifting organic molecules out of a non-living environment, whether that means ingesting organic molecules in the soil, filtering them out of seawater, or some other source where you're not taking it directly from another living thing. But one way or another, you as an animal must take in external organic molecules to feed yourself, to power your metabolism. Now, we are not the only heterotrophic organisms. A great majority of Protists are great majority of protists are heterotrophic. A great majority of prokaryotes are heterotrophic. We are not the only heterotrophic multicellular organisms even. In fact, the fungi, kingdom fungi, are also heterotrophs. And I don't know if I've said this in here before, but genetic studies show there is a closer relationship between animals and fungi than there is between either group and plants. So if we're sketching out a family tree of life, we've got the plants going off one way, and then a two-branched additional pathway with fungi and Animalia more closely related to each other than either is to the plants over here. All of them stem from the kingdom Protista. That is not in dispute, that the group of organisms today loosely classified as the protists were the first eukaryotes and the source of all multicellular diversity. But plants share this feature of, fungi share this feature of heterotrophy in common with animals, and it's believed that fungi and animals had a common heterotrophic protist ancestor. Now that said, animals do heterotrophy a bit differently than fungi, namely that as an animal, you have internal digestion, meaning that you process your food inside of your body, that you have compartments of some kind in your body where the food is broken down, that the majority of animals have some kind of internal gut or space within the body in which food is processed. There are a couple of weird exceptions. Every rule in biology has an exception. You all know that by now. There are a couple of animals that engage in very explicit external digestion. For example, a lot of spiders will inject digestive enzymes into the insects they prey upon and then suck up the nutritive fluids afterwards. But even then, that nutritive fluid has to be taken into the animal's body in an internal gut. So not only heterotrophy, but heterotrophy with internal digestion is a defining feature of being an animal. Anything else? A few things. Let's talk about cellular structure. Obviously, animals are eukaryotic. That's already been well established. We have cells with nuclei. Animals are multicellular. There are no single-celled animals. There are some microscopic animals, but no single-celled animals. But in terms of our cellular structure, we hold those cells together differently than other forms of multicellular life. Both fungi and plants are bound together as a result of cell walls, that they have compounds in their cell walls which cement nearby cells together, joining them to each other. In plants, the cellulose cell wall binds plant cells to other nearby plant cells. In fungi, we have a cell wall made of chitin, which does the same thing. Not so in animals. One of the most distinctive features of animal cells, as compared to every other form of cellular life on Earth, is that animal cells are the only cells that lack cell walls. Every other form of life, even prokaryotes, have cell walls. Animal cells do not. We lack that special shaping barrier around the outside of the cell. As such, we can't join animal cells together by connecting their cell walls to each other like plants and fungi do. As a result, animal cells are instead held together as a result of special structural proteins and glycoproteins, the most common example being extracellular collagen, C-O-L-L-A-G-E-N. Collagen is the most common protein in the bodies of animals. In your body, collagen is about 25% of all of your total protein. And collagen is a vital feature of most forms of animal life, that we have extensive extracellular matrix, partly proteins, partly fluids, and the extracellular fibers bind together animal cells, collagen being the most abundant of those. In addition, some animal cells are joined together by a variety of multicellular or intracellular junctions, as I should say. There are a variety of connections that can form between cells, holding them together. You might think back to your Bio 1, things like tight junctions and gap junctions. These are examples of ways in which nearby cells can be bound together as part of the same tissue. Speaking of, right there is another distinctive feature of animal body structure, the presence of tissues. Now, we use the term tissue with regards to plant and fungi in a more informal sort of way. We talk about, for example, vascular tissues in plants, which are those cells that are specialized to draw water and nutrients through the plants. But when we talk about tissues in animals, this is a defining quality of the structure of animal bodies, that our cells are organized into distinctive tissues. A tissue being defined as a group of cells with common features that are working together for a common function and are derived from the same part of the embryo. So tissues are groups of cells with related functions coming from the same embryonic origin. Tissues are a defining quality of animal organization. All but the simplest animals possess true tissues. There's an animal called a sponge. We're going to talk a bit about sponges. You've already seen a video about sponges. But the phylum Porifera, the simplest and most primitive group of living animals, do not have true tissues. But they are the exception, not the rule, and they represent a very primitive representative of early animal life. All other living varieties of animals have tissues. Furthermore, animal tissues contain a much wider variety of types of cells than those of other forms of life. You have more than 250 known types of cells in your body. And animals have types of cells with no analog in any other living thing. You have epithelial cells. Plants and fungi have something like epithelium, like forming outer coverings. You have connective tissues or something like them. Plants and fungi have tissues like that or something like a connective tissue to bind structures together. But animals have two tissue types that have no comparative form in other forms of multicellular life. Namely, animals have muscular tissue, meaning we have specialized tissues for contracting to generate movement, and we have nervous tissue, specialized tissues for transmitting and receiving coded electrical information. These are things that animals have, again, except for sponges. Plants and fungi have no comparable cell or tissue-like types. Brings us to another feature of being an animal, motility. Motility, M-O-T-I-L-I-T-Y. If you don't know that term, motility is ability to move. Animals are the only multicellular life that can move under their own power. Again, you get weird exceptions. Slime molds among the fungi can move at one point in their life cycle. But animals are unique in that all animals at some point in their life cycle are capable of independent movement. They're capable of moving under their own power. I should point out, not all animals stay that way. All animals have a motile stage of some point in their life cycle, and most animals remain motile throughout their lives. But some animals lack the ability to move after a certain point in time. Some animals will have a larval form which is capable of movement, and then they'll settle someplace, anchor to a substrate like a rock, and never move again. Animals that do not move around are known as sessile animals. S-E-S-S-I-L-E, sessile. Think of barnacles, for example. You know, the barnacles that grow on the side of a ship or on the side of a dock. Those are sessile varieties of animals that have planted themselves in one spot and do not move around anymore. So motility at some point during life is a key animal feature. And speaking of animal life cycles, let's talk about the animal developmental cycle, because that is another feature of being an animal that is quite unique, that no other living things exhibit anything like it. First off, most animals reproduce sexually. Sexual reproduction is the default reproductive mode among animals. That is, you have large, non-motile eggs produced by the biological female, which fuse with small, motile sperm produced by the male. That's the default reproductive model for animals. There are a few forms of asexual reproduction among the Animalia, but they tend to be sort of opportunistic forms of reproduction. For example, the fragmentation reproduction that we see in a starfish when one of its limbs gets cut off and grows into a new starfish, or the budding reproduction that we see in some very simple animals like hydras, or parthenogenesis, which some of you might recall from Bio 1. which is that situation in which, in the absence of a male, the female has a way to fertilize her own eggs by duplicating their chromosomes. But these are all, again, weird exceptions, and for the most part, they are occurring in species that otherwise reproduce sexually. Even sponges exhibit sexual reproduction. Now, once sperm fuses with egg, that forms a single diploid cell called a zygote, Z-Y-G-O-T-E. Now, this is important because another feature of animal development is that we do not exhibit the alternation of generations. Think back to your understanding of plants and fungi. Plants and fungi both exhibit alternation of generations, in which they have a haploid generation, like the gametophyte in plants produces haploid gametes, which then fuse together to make the diploid sporophyte plants. So the sporophyte then makes haploid spores, which form into gametophytes without being fertilized. So if you're a plant, you exhibit alternation of generations. Some of your organisms will be haploid, meaning they have one copy of each chromosome. Some of your organisms will be diploid, meaning they have two copies of each chromosome. Most fungi are like this as well. Not so with animals. Virtually all animals are diploid. We have a few weird examples of tetraploid animals, but diploidy is the normal configuration for animal life, meaning that animals typically have paired chromosomes, that they get half of them from mom, half of them from dad. The normative reproductive model for animals is you have diploid parents, male and female, which produce haploid haploid gametes, that is gametes, eggs and sperm that have only one copy of each chromosome. Egg and sperm fuse into a diploid zygote. And as a result, the offspring are diploid just like the parents were. So you've got diploid parents make haploid gametes to make more diploid offspring. There is no such thing as a haploid generation of animals running around. Now, not only do animals exhibit commonality in their genetic structure and their diploidy, but also in the sequence of events that happen after fertilization. Egg fuses with sperm, and then you get, of course, a single diploid cell called a zygote, Z-Y-G-O-T-E. After forming, the zygote begins to undergo cleavage, begins dividing mitotically, forming a ball of cells, 2 cells, 4, 8, 16, gradually assembling this sphere of cells, a multicellular ball called the blastula, B-L-A-S-T-U-L-A. The blastula is going to be the basal form that we all start as. We all begin as a ball of undifferentiated cells. Now, once that blastula gets large enough, it undergoes gastrulation, G-A-S-T-R-U-L-A-T-I-O-N. Gastrulation means that ball of cells folds inward on itself. It begins to indent and invaginate. If you don't remember that term from previous classes, invagination is infolding of a tissue or of a membrane or what have you. So my blastula has begun to gastrulate, folding inward on itself, and this forms a multi-layered hollow sphere with a single opening to the outside. Let me draw this for you so that you can get an idea what I am talking about visually. All right. And eventually what you will have, again, is this kind of hollow, multi-layered ball of cells called the gastrula, G-A-S-T-R-U-L-A. So you can see what we did here. We've got the blastula as just a hollow, undifferentiated ball of cells on your left, and you can see we undergo gastrulation, in which that thing begins to pocket inward and invaginates, until eventually you end up with this two-layered ball of cells called the gastrula. Prefix gast means stomach, so it's kind of like formed a stomach. It's formed an internal space, right? That little opening, incidentally, that we see there on the gastrula, that little tiny primal opening, is called the blastopore, B-L-A-S-T-P-O-R-E. And that is going to eventually become one of the external openings to the body. Which one depends on what animal we're dealing with. That cavity that we have formed within there, that primal cavity, is called the blastocele. B-L-A-S-T-C-O-E-L, blastocele. That word, that prefix seal, C-O-E-L, you're going to see that a lot when we're talking about body cavities. It refers to a cavity or a space inside of the body of an animal. All right, so that's our initial developmental pathway there. We've got blastula, gastrulates, folds inward on itself, becomes a gastrula with a blastopore and a blastocele. Following that, we begin to experience more complex development. We begin to experience differentiation, that the cells are going to start to differentiate at that point. And if you don't remember that term from Bio 1, D-I-F-F-E-R-E-N-T-I-A-T-I-O-N, differentiation. Whenever cells differentiate, that means they are assuming specific cellular identities, that they're ceasing to just be stem cells, and they're becoming more specific kinds of tissue cells. Once we have differentiation, that gastrula will form distinct layers of cells that we refer to as the embryonic tissues, or sometimes as the germ layers. Depending on what kind of animal we get, we'll have either 2 or 3 of those. More on that in a bit. At this point, our gastrula begins to become an embryo, E-M-B-R-Y-O, which is, of course, the primordial form of the animal in which cells are beginning to assume specific functions and identities. Some organisms, not all, exhibit a stage of development between gastrula and embryo called the nerula, N-E-R-U-L-A. In the nerula stage, you're going to get a band of tissue that forms along the length of the gastrula called the neural streak, N-E-U-R-A-L. The neural streak is eventually going to give rise to the nervous system in animals that have a central nervous system. So nerula stage is part of most animals' development, not all. Embryonic development, once you get there, little by little, the organism is going to assume more and more identifiable features. Initially, as you've already seen from our early embryology in this class, that not going to look a whole lot different than the embryos of other animals. If you look at every animal in its earliest embryonic stages, they all look basically the same. As you get a little further on, it's going to get more and more recognizable qualities until eventually, for example, all vertebrate embryos have a basic model that they all look like, and all mammal embryos have a basic model that they look like, and eventually all primate embryos have a basic model that they look like. So you can identify a little bit more of what's going on at each stage of this development. But you spend a lot of your early development basically as an undifferentiated wad of cells. For the first month or so that you exist, you don't have a heart, you don't have a brain, you don't have bones. What will eventually become your limbs is nothing more than a small bud. So you would not really look at that thing and say, yeah, that's a human being. Really, at this point, all animals look basically the same. Now, what happens next here depends on what kind of animal we are dealing with. We all have those stages that go up to embryo. In some animals, we're going to experience an early stage of development in which the embryo develops into a form of the animal that is not yet its adult or final form, that you're going to have an immature form of the animal that is not reproductively competent yet. We call this organism a larva, L-A-R-V-A. So a lot of animals exhibit a larval stage of development in which they give rise to this sexually immature form that does not yet have all the features of the fully developed adults. The purpose of the larval stage, more than anything else, has to do with energy conservation on part of the mother. That in most animals that lay eggs, we have the situation whereby mom is producing large numbers of eggs at a time, by which I mean thousands of eggs at once in many cases. When you're an insect or a small invertebrate, or even a very simple vertebrate like a frog, your whole reproductive strategy consists on producing hundreds or thousands of tiny offspring at once in the hopes that a couple of them make it to adulthood and survive. Well, if you're producing thousands of eggs at one time, mama animal cannot invest all that much energy in every individual egg. So the solution to that developmental problem is not to give the young everything that they need up front. That basically mama animal lays an egg that hatches into a partially developed form. And that partially developed form, that larva, basically devotes itself to eating until it has enough fuel to power its growth into a more complex form. When that happens, the larva will go into a hibernating state. Often it will form a sort of cocoon called a chrysalis, C-H-R-Y-A-L-S-I-S. Not all larvae have chrysalis stages, but most do. But the animal will experience, whether it has a chrysalis or not, the animal will experience a developmental resurgence at some point that we call metamorphosis. S-I-S, metamorphosis. In metamorphosis, the animal has a secondary stage of developments beyond its initial embryonic developments. During that metamorphic stage, the animal's body undergoes a radical reorganization. Its anatomy changes significantly into a more complex form. Sometimes that more complex form is the adult form. Sometimes that more complex form is another larval stage. Some animals undergo as many as 4 or 5 distinct larval stages before they hit the final adult phase of their existence. In a lot of insects, multiple larval stages are the norm. Now, metamorphosis, don't think of this being like a Pokemon that suddenly one day it starts glowing and you're like, oh, look, my caterpillar's changing, and bam, it turns into a butterfly. Metamorphosis is going to be a slow process. Sometimes the animal goes into hibernation, cocoons itself in our chrysalis during that process. Other times, that animal is going to undergo a more gradual metamorphosis during which it remains active. Think, for example, of the transformation of a tadpole into a frog, that during its metamorphic cycle, it remains active. Slowly, it resorbs its own tail. Slowly, it grows limbs until eventually you have a frog that doesn't look a whole lot like the tadpole that it was and that's capable of surviving on land. So metamorphosis can be gradual. It can be sudden. You can remain active during it. You can go into a chrysalis during it. But even quote-unquote sudden metamorphosis takes time. It's going to take periods of days or weeks for the animal to undergo those complex cycles of change. Now, not all animals do that. Not all animals go through a situation in which the mother has minimal investment in the offspring and you produce lots of them with the knowledge that most of them are going to die in the hopes that a couple of them make it to survive. An alternate reproductive strategy that many animals exhibit is to have smaller numbers of offspring with higher investment of maternal energy, in which case the offspring has a longer gestation period, whether that means in an egg or in mom's body, that offspring is going to be essentially fully developed at birth, that it will go through a fetus stage, fetus, in which you basically have a miniature form of the adult that when it's ready to function outside of the egg or the womb is going to emerge as basically a small adult. That the organism will look in childhood like it looks in its adult stage. That's pretty much what that is going to mean. We exhibit this reproductive strategy in which we produce far fewer offspring at one time, and they are much more fully formed at birth. Mammals in general, this is how we do things. But these are some common developmental patterns. I should point out all of this is regulated by a common set of shared developmental genes, often called Hox genes. And when you write Hox, you capitalize it, H-O-X. You also italicize or underline it. That's standard convention for naming genes. Hox genes are a family of genes that regulates the process of development. They're genetics that are not really terribly involved much after your birth. They're involved in handling your developmental stages. They're involved in your reaching puberty and things like that as well. But a significant amount of your Hox genes are only active during your embryonic development, making sure that you end up with limb buds, and lungs and a head that's distinct from the rest of the body and all of that sort of stuff. So that is going to be our basic genetic developmental sets. Hox genes are actually some of the most strongly conserved genes in animal evolution. The Hox genes in very primitive animals have definite similarities to those found in you. And for closely related animals, most of the genetic difference between you and them is in their Hox genes. Most of the genetic differences between us and chimpanzees, which you might recall chimps share 99% of our DNA, and most of that 1% that they don't share is differences in our developmental genetic sets. Incidentally, possession of a complex set of Hox genes for the regulation of development is a really important feature of being an animal. And having studied Hox genes, we're learning an awful lot more about how animals have evolved and how they are related to each other. Because they're so strongly conserved across animal groups, studying changes in Hox genes is a very important clue for looking at how animal development works. We actually talk a lot these days about a sort of subdiscipline of biology that is sometimes called EVO-DEVO, E-V-O-D-V-O. EVO-DEVO, that's for evolution and development. Looking at the developmental process as an indicator of how our evolutionary heritage works. We've been doing that since the 19th century. A German biologist by the name of Ernst Haeckel, back in the 19th century, conceived of the idea of ontogeny recapitulates phylogeny. I think I said that earlier this semester whenever we said that it was believed for a long time that an animal went through all the early stages of its evolutionary development during its embryonic development. We know that's not exactly the case, but we also do know that you can look at embryonic stages of animals to learn things about their evolutionary history. And EVO-DEVO is all about that, and Hox genes really help. So this gives us a basic slate of things that we can use to identify and say, that critter right there, that's an animal. This organism is an animal because it exhibits diploidy with sexual reproduction in every generation. This organism is a multicellular eukaryotic heterotroph with internal digestion. This organism is going to lack cell walls, but it's going to have extracellular proteins for binding it together. It's going to have true tissues, including muscular and nervous type tissues. It's going to exhibit motility at some point in its life cycle. And it's going to exhibit a distinct developmental process, which may or may not feature larval forms, and which is regulated by Hox genes. These are the defining qualities that make an organism an animal. And anything that does that, however weird or primitive looking you might think of it being and say that thing doesn't look anything like an animal at all, like a sea anemone or a coral, for example, but it does all those things, it is an animal. All right, sounding good? Before I go on, this actually seems like a good time to mention a set of developmental theories that are not just for animals, but for life as a whole, that when we're talking about multicellular life, we talk about reproduction in terms of species being R-selected or K-selected. Lowercase r selected species are the ones that have low parental investment in large numbers of offspring. In other words, there's not a lot of energy invested per forming each offspring. They very often exhibit larval stages, but not required. They have large litter sizes in the hopes that a few organisms will manage to eke out survival. Mice, for example, they don't have a larval stage, but they do exhibit R selection. They have large numbers of offspring because most of them are not going to make it. By contrast, a K-selected species, and that's capital K, is one in which there is a high parental investment, both pre and post-birth, in a much smaller number of offspring. We are a K-selected species in that we have a much smaller litter size, that the offspring are basically miniature adults at birth, and that there is a prolonged period of parental care. Now, there are some problems with this model. There are a lot of exceptions to it, but it's a good way of looking at reproductive strategies in general, and you will find nearly all animals fall into one or the other of those patterns clearly, where it's like, let's make lots of offspring with low parental energy investment, or a few offspring with high investment. And by the way, that terminology R versus K is algebraic terminology. The R and K are algebraic terms that come from fairly standard equations used in the field of ecology, but we're not going to get into their specific meaning beyond that. Okay, so that's how we define an animal. How long have these things called animals been around? Thought I was going to give you a nice, neat answer to that. I wish I could. The entire field of zoology wishes that we could. And by the way, Z-O-O-L-O-G-Y, it is zoology, not zoology, the study of animals. Now, zoology as a discipline has not yet come to a clear agreement on exactly when the Animalia as a group first appeared. We have not yet fully sorted that out. There have been suggestions pushing the origins of Animalia back all the way to 1.5 billion years ago. That is, I must say, kind of dubious at this point. What we have from that far back are trace fossils of basically blobs of cells, and very little that would tell us whether the blob in question was a primitive animal-like organism, a primitive fungus-like organism, a primitive plant-like organism, or for that matter, just a colonial protist. More commonly suggested is an origin for the Animalia going back approximately 1 billion years. There are some trace fossils that look like they would indicate trackways made by certain simple microscopic animals today. But even that is dubious because similar trackways are made by certain kinds of protists as well. The first definitive fossil animals, well, the first possible fossil animals, let me just go with that. The first possible fossil animals are some fossils that resemble sponges coming from a formation, a rock formation called the Trizona. The Trizona Formation. It's located in southern Australia. The Trizona fossils date back about 665 million years. And they look kind of like sponges, but again, hard to tell if they might be fungi or something. Mitochondrial DNA suggests to us that all animals share a common set of genes inherited from a common ancestor going back approximately 650 million years. So not necessarily the first animal, we all know how that works by now, but it does seem that all animals currently living can trace their origins to a common ancestor 650 million or thereabouts years ago. Even so, though, we can't guarantee that that common ancestor was necessarily an animal as we understand it right now. That it wouldn't even necessarily have anatomical features that we would say, yep, this thing is an animal. The oldest definitive animal fossil right now is an organism called Dickinsonia. Dickinsonia. That is a genus name, so italicize it or underline it and capitalize it because it's a genus name. Dickinsonia is a whole genus of animal life. The specific oldest confirmed Dickinsonia fossil is of a species called Dickinsonia costata, Costata. Italicize or underline that, but do not capitalize it. It's a species name. So Dickinsonia costata appears to be a very primitive animal that lived in what is now Russia. The fossils are from northwestern Russia, going back about 558 million years. Like so many of these early fossils, it appears to be basically a blob. It's not really got a lot of distinct anatomy, but we have looked at this fossil chemically and discovered that Dickinsonia, among other things, Dickinsonia costata, contains cholesterol. And cholesterol is a specific animal lipid. So we know that Dickinsonia was a primitive variety of animal. A lot of textbooks will tell you that the earliest possible animal fossils go back to a group of organisms called the Ediacaran fauna. Ediacaran. Ediacaran. Capitalize that name. It's the proper name of a geologic period of history. Fauna, Fauna. It's a general term referring to animals. So the Ediacaran fauna are the fossils of animals from the Ediacaran period. The Ediacaran fauna all appear to have been very simple animals by modern standards. There's a lot of ambiguity in these fossils. There's a lot of debate over whether they are animals or a kind of plant or a kind of fungus. But among the Ediacaran fauna, we have found fossils that resemble modern corals, fossils that resemble soft-bodied mollusks, and fossils that resemble primitive worms. The Ediacaran fossil beds are from Australia, and they date to somewhere in the neighborhood of about 610 to 660 million years ago, depending on which fossils you look at. The Trizona Formation is an example of Ediacaran fossils from that time. So the combination of genetic and fossil evidence strongly suggests that there were animals, as we understand them now, somewhere between about 600 and 650 million years ago in Australia, or what's now Australia. Trizona Formation means it could go back a little bit further than that. Some other dubious fossils suggest the possibility of animals as far back as 1 billion years ago. But the oldest confirmed animal fossil that we can say, yep, that thing is definitely an animal by our modern definition, is Dickinsonia 558 million years ago. Now, whatever our starting point was, the Animalia branched off from the protist. Just like plants and fungi, animals started from simple, single-celled eukaryotes. Much like with plants, there's a lot of argument over where exactly to draw the line between animals and organisms that are sometimes called protozoa, Protozoa, the protozoa being animal-like protists. That is, protists that are motile heterotrophs, and that in many cases have sexual reproduction. The general agreement is that the closest living relative to the kingdom Animalia among non-animal species is a group of protists called the choanoflagellates, Choanoflagellates. Choanoflagellates. Choanoflagellates are still around today. They are a group of colonial flagellated protists. That is, they are protists with flagella, whip-like motility cells located on them, motility fibers. Colonial in that they are single-celled organisms, but their habitual way of living is to band together in clusters or colonies for mutual protection. Colonial protists would be the origin point for our more complex forms of life. You had to have single cell at some point, and at some point single cells needed to begin cooperating with each other. It appears that what happened is that you ended up with spherical colonies of choanoflagellates or protists like them. We see them doing that today. Now, the cool thing about that is the colony is mutually protective, right? Cells get defense from other cells around them. A big group of them is harder for larger predators to go after. They can provide mutual supports. In some cases, they can even recycle the byproducts of one another's chemistry for mutual efficiency. Now, here's the thing. If I've got a whole group of cells that are living together in groups, selective pressures are going to favor adaptations that make you more effective as a group living cell. In other words, they're going to get better at working with each other after a while. Among other things, that's going to mean that you're going to begin developing certain regions of the colony that are better at things than other parts of the colony, which the rest of the colony benefit from. Like maybe there's a patch of cells over here in my choanoflagellate colony that is better at metabolizing a certain nutrient. And that's great because now it can pass on the products of that to other nearby cells. Or maybe you've got some of them that have tougher cell walls because the ancestors of animals were protists that did have cell walls. And it's like, hey, these will make a good outer layer for the colony. Or maybe you'll get some that produce protein fibers, which can bind other cells together so you don't need a cell wall anymore after a while. Point is, natural selection is going to favor adaptations in my colonial protists that make them better at functioning in groups. And that means adaptations that basically let individual cells do things that benefit the rest of the colony for their own purposes. Now, as you get these colonies getting larger and more stable over time, you get dedicated regions of the colony that have specific features that make them better at doing specific things. Symbiotic relationships develop in which parts of the colony become dependent on other parts of the colony for carrying out mutual functions. Horizontal gene transfer among them means that genetic fragments get passed back and forth so common adaptations get shared throughout the colony. The real moment of truth, after you've got extracellular fibers binding these things together and you've got regional specialization in different kinds of things, is going to be the development of specialized reproductive centers. That is, populations of cells within the colony whose primary function is the production of gametes for the colony as a whole. And those gametes can then pass on this constellation of features to the offspring. Once that happens, once we've got certain cells in the colony being responsible for regulating the reproduction of all the other ones, and you've got specialized groups of cells doing different things and bound together by extracellular protein, at that point, we have essentially a multicellular organism. It's no longer meaningful to talk about these things as being individual single-celled organisms in a colony. They have become a truly interdependent collection of cells that rely on this multicellular arrangement for their continued survival and reproduction, and that carry out specialized tasks for the benefit of the colony as a whole rather than just for their own success. That's multicellularity right there, folks, and that is where we have something that ceases to be a colonial protist and becomes a true animal. And if this all sounds a little bit unlikely, I would suggest that you go into the Blackboard articles folders, and you will find an article there about how modern experiments with multicellular yeasts that are colonial protists, placing them into agitated or stressful environments, they can begin forming multicellular independent relationships in as little as 6 months. So this is definitely a plausible thing to have happened. We've watched it happen under lab conditions. It's certainly a thing that could have happened in the early animal past. So this is our dominant model for the origins of the Animalia. We've watched it play out. We've watched existing colonial protists develop specialized patches of cells that do particular things. It's not a big jump. So that is where we get our origins. Now, our initial animal doesn't look a lot different than a colonial protist. It's basically a blob. We sometimes call them proto-animals. These primitive animal-like structures that are animals in name only, that are basically checking the boxes of most of the features that we associate with living animals, even missing one or two of those, however, like missing true tissues as well as muscular and nervous cells, at least initially. But over time, those features are going to begin to develop. Our proto-animals are going to look an awful lot like the most primitive living animal phylum today, the phylum Porifera, capital P-O-R-I-F-E-R-A, better known to you as the sponges. Sponges are exceedingly primitive animals. They're sometimes called the basal animal, and you will find that a sponge is an exception to a lot of our basic rules of how we expect an animal to look and behave. Sponges are sessile, although they have a mobile embryonic stage, a mobile larval stage. Sponges do not have true tissues. They do not have muscular or nervous tissue. They don't even have symmetry. But a sponge gives us a pretty good picture of what the very earliest animals looked like, and they have remained a very common group of animals in the environment today. So that's our origin. Got a lot more story to tell here, but I think I've given you enough to chew on for the time being. In our next lesson, we are going to take a little bit more look at the history of the Animalia, talk about how animals have developed and changed over time. You can tell this is going to be a fun lesson because it's the lesson where I get to tell you about dinosaurs. Everybody loves dinosaurs, right? So next time, animal origins with some special attention to our buddies, the dinosaurs. And then from there, we will move along to looking at some of the more specific features that set animals apart, including details of their body plans and how they develop. Sound good? All right. Hope you're all having a great day, folks. I'll talk to you later.